Happy Tuesday, Limbrook Bio students, and welcome back to Chapter 10. This starts the last section of our year together. Genetics leads to some combination of reproduction and heredity, and eventually we'll be able to talk about evolution. Some of you began this material for a day or so before we all left, but I skipped most of that stuff here, so it should be mostly new material. As I said back then, this first part is kind of a throwback to the start of the year when we skipped talking about nucleic acids because we'd get to it eventually. So now is the eventual time. Looking ahead briefly, this chapter will take about 2 weeks since we're only doing 2 days of work per week now. Because of that, though, some of these videos are going to be longer than we've been doing. Today's is definitely going to be a little bit longer because I'm putting a few clips in, and we're going to plow ahead through some of the intro things. So I want to just make sure we get through everything. Also, this topic may get confusing quickly. Hopefully not until next week, though. So you can't hesitate to ask any questions and use the virtual extra help in Microsoft Teams. Material can get away from you fast, and then the rest of the year just kind of spins out of control. So on that cheery note, let's dive right in. Nothing to see here. Today we're going to go over the basics of genetics, what DNA looks like, how we know this, and how we can make more DNA if needed at some point. A good amount of information, but really nothing to worry about. I want to try to get through this beginning stuff as quick as we can, just because we've seen a lot of it before, and then that leaves us plenty of time to go over the more, not complicated, but more important things later on. So just some stuff that hopefully you remember from the beginning of the year. All living things have to have DNA in their cells. DNA is the instruction manual for making proteins, and we are mostly made up of proteins. We have fats and sugar and stuff in our bodies, but protein are what mostly is the structure of us. And if you remember, DNA stands for deoxyribonucleic acid. That's what those letters stand for there. Now, I don't think we've really gone into this much since the beginning of the year, but in this whole big DNA molecule right here, that large model I've got in the classroom, a gene is just one section of the entire instruction manual. So if you hear someone say the word gene, or you've ever used the word gene or seen it in class at some point, genes are just little single recipes for things. So if we think of our big entire DNA molecule as a giant recipe book like this, think of the biggest, most complicated recipe book anywhere on the planet. A gene would be like one single recipe for something. It may only be one page, it may be a single paragraph even, but it's just one little section. So like on this molecule here, maybe this little part right there, that's a gene. Maybe down here, that's another gene. This one up here maybe is the hair color gene. This one is the eye color gene. Say a single little section of a much, 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 much larger entire DNA molecule. And genes are what make us who we are. Inside and out, who we are in our personality, what we look like, things in our bodies that we can't see, what's going on inside our bodies, everything about us is controlled by our genes. And as we've seen a little bit of at certain times, mutations are these random changes. They're differences. They're sometimes kind of like mistakes, basically, of nature, stuff that wasn't expected, but it's not necessarily bad. But mutations are why we're so different, why we don't all just have the exact same height and eye color and hair color and everything and just be essentially clones of each other. Mutations are those changes, and they can be really good. So again, going back to stuff from the beginning of the year, DNA is a nucleic acid. So it's made up of these units called nucleotides. What are the parts of the nucleotides? We only said about 30,000 times in October or so. Remember, what are the parts of nucleotides? That's right, phosphate, sugar, and base. So phosphate, sugar, and base are the 3 parts of a nucleotide. So this whole thing right here on the bottom left, that is one nucleotide. That's the building block of a nucleic acid, of which DNA is an example. RNA is also a nucleic acid type. We'll see that tomorrow or Thursday for you when you're looking at your videos. And there are 4 different types of bases. As you can see right here, each one of these things is a nucleotide. So here is one nucleotide. This is one nucleotide. This is one nucleotide. This is one, and so on and so on. You see 2 different chains. One seems to be kind of going this way, if we see sort of where the point is facing, and this other side is kind of going the other direction. So they run sort of opposite to each other. But DNA is going to be made up of this. It's going to be 2 opposite-facing strands of nucleotides, 2 little chains of nucleotides that just sort of link up with each other in the middle. So if I just erase this ink right here, you can see all these 4 different types of bases. There's one type, there's another type, there's another type, and there's another type. They all have different letters that signify what base they are, and we'll see that in a little while, what that means and what these different types of bases actually are. But this is the basic structure of DNA, and we're going to go into this a little bit today mostly, and then do just a little bit of stuff you'll actually be able to practice and do some work with. The famous shape of a DNA molecule is what we call a double helix. So it's kind of like a ladder. Think of, again, that molecule, the big model I have in the front of the classroom, and how you can stretch it out to look kind of like this, just sort of a big ladder with these steps in the middle of it. But then when you twist it up, it just becomes this. It's just a twisted staircase. Or a ladder. That's all it is. And you oftentimes hear it called a twisted ladder. But it has those same pieces we've been seeing. Here's your phosphate, here's your sugar, and here's your base. I did those kind of out of order. But phosphate, sugar, and base. So all together, they are right there. Phosphate, sugar, base. Here's another phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. And they all just link up like that. So that then gives you this long chain of these things. Phosphate, sugar, base. Here's a long chain of phosphate, sugars, and bases on one side, and now another chain of phosphate, sugars, and bases on the other side. They attach to each other to make this big ladder, and as you can see now, again, then you twist it into this big thing you call a double helix. It's basically a big corkscrew kind of shape, but you call it a double helix because you've got the one chain right here, and you've got then another chain right here, and they just sort of twist around each other and intertwine in that way. Now, in each of our cells in our bodies, we have between 6 and 9 feet of DNA. That's insane. It's just incredibly tiny and tightly packed. It's just remarkable how these molecules get all kind of twisted around each other to fit inside every one of your cells, but there's a lot of DNA, and that's the complete instruction manual for making you. So every bit of information you need to make a complete one of you is in each of your living cells. And to figure out the shape, this is a major thing. This is not something we've known for relatively that long. This is really a big deal and one of the most significant discoveries of all time. And that's mostly what we're actually going to look at today. We're going to take it slow this first time today and get into that kind of stuff. If you ever forget what the molecule is called, you can just actually refer back to this cartoon with this very scientific description here of a long, twisty, rope laddery type thingy. Perfect description right there. So if we were all together in the classroom right now, I'd be taking your picks and your guesses as to who actually made this discovery. You may know already. If you do, keep it to yourself. Don't tell your classmates. I know that's not too difficult right now, but let's say it's the early 50s. Everyone in the scientific community wants to know what DNA looks like. That we've come to this conclusion that DNA is the molecule that's going to carry information about us and be passed on from parents to children. And no one really knows much about it. We know it's there, but really not a whole lot about what it really is. So there's some major minds in the scientific community at this point trying to solve this question. And I want to think or see who you think actually did this. So here are our choices. Our first contestant is a British chemist who's already pretty famous at this point. They are an expert at using X-ray pictures. You can get X-rays to look inside someone's body, as you've probably done if you've ever broken a bone or anything like that. But you can actually use X-rays to get these really cool detailed pictures of molecules we can't see with the naked eye. So this person was already an expert in this field and already used these things to find the structures of tiny little things like viruses, and got actually pretty famous for doing that with a certain type of virus. The next option is a pair of scientists. So one's a very young American biologist who liked birds from Chicago, and the British friend who wasn't actually a PhD, never got his doctorate because he had been working on a project for a couple of years, and then all of his research, all of his work got destroyed during World War II when a bomb fell through his roof in London and destroyed everything he'd been working on. So he just never bothered to finish and kind of moved on with other material. And our third option is an American biochemist who to this day is still one of only 4 people to ever win multiple Nobel Prizes. And this actually person did in multiple fields. That's pretty impressive too. And actually wrote the book on protein structures. And you hear people say, oh, that person wrote the book. This guy literally wrote the book on what proteins are structured like, and was later named by, I forget what magazine or newspaper, as the 16th most important scientist who ever lived. So who's your pick? And the winner is, drum roll please, Francis Crick and James Dewey Watson. That's Watson in the lower left. He's about 25 here, so a pretty young guy. Crick was much older. He's pointing to their original tin model of the double helix. And here they are as bobbleheads. Notice Watson's famous bucket hat, for which he just is very fond of to this day. What happened is, this guy named Maurice Wilkins, who was also working on this project at a competing laboratory in England, he let Watson see a photograph taken by a researcher of his named Rosalind Franklin. She was the one who was the expert in X-ray diffraction, and she worked for him, but this wasn't really as ethical as it could have been to show your employees' work to someone else who was competing against you. They really were the competition, and she had no idea this had happened. So anyway, Watson saw this picture, the little X picture there. This is a famous picture at this point. And he knew that an X pattern meant a helix. Crick was someone who knew some stuff about X-ray pictures and had told Watson this. So as soon as Watson saw this picture, he started just basically losing his mind. He knew this was a big deal. He made some notes for himself on the trek back to his lab, and it really didn't take the two of them very long at that point to figure out the structure of the molecule, unfortunately. So that was Jim Watson telling his story as part of a documentary for PBS to mark the 50th anniversary of the discovery of the double helix in 2003. But what about everybody else? Well, the last thing I said in that last slide was they made the discovery unfortunately. And that's because Rosalind Franklin died pretty young. It's not clear if she ever knew how much of her work led to the discovery of the structure. Watson and Crick shared the Nobel Prize with Morris Wilkins, but the award is not awarded posthumously, so it's not given to someone for their contributions after they've already died. So she was never recognized, and it was decades before her contributions were really known and recognized by the scientific community. For this, she is often called the Dark Lady of Biology, and there have been a couple of books and even a stage play pretty recently about her story. As kind of a sad aside here, one of her notebooks contains a brief note to herself speculating that DNA might have a helical shape. So it's pretty likely that she would have actually made the discovery herself, given enough time. And she seemed like she was pretty close. She should have probably been the favorite at this point, at that point back then. As for the other guy, the American Linus Pauling, he was very famous and just kind of had to make do with that. He basically accomplished everything but discovering the double helix. He was a prolific writer, as you can see. He wrote a bunch of books and papers, I mean, in the thousands. He really was a worldwide celebrity, as famous as people like Neil deGrasse Tyson are now. He's very accomplished for himself, but he's more of a science communicator, an advocate, and an ambassador for science, where Linus Pauling was making really groundbreaking discoveries and doing really great work, and was known worldwide for this stuff. And like I said, he's still to this day one of only 4 people to ever win multiple Nobel Prizes, and one of only 2 to win in different fields. I have no idea who Robbie Kraft is, but I love this tweet, and I'm just going to leave this right here. That's another clip from the 50th anniversary documentary in 2003, and you may have noticed Watson wearing his bucket hat in it. I worked at Cold Spring Harbor a decade later in that white building on the water at the start of the clip, so I was there for the 60th anniversary symposium and took these pictures of Jim. I love this picture on the left with him standing in front of a giant painting himself, because the best part right here is that the artist actually painted the bucket hat at his feet in the picture. I love that little detail there. He always had it with him and always on his head, but he took it off for the painting, and I guess he left it there and they decided to put it in. Somewhere I have my commemorative bucket hat that was given to everyone who attended the big garden party on the day this picture was taken. And I'm not proud to say that I swiped mine off of an unattended chair, but I actually am kind of really proud of that, because the hat's an awesome memento that you're never going to find anywhere else. So usually when I watch the full video with classes, I usually watch the entire documentary. Kids comment that Watson seems a little bit out there, is the nice way to put it. I point out that he was about 70 when this was made, and that he's really only gone downhill since then, unfortunately. In fact, he turned 92 within the last week. At the time I'm recording this, I think it was about 2 days ago that he turned 92 years old, which good for him. But to be clear, this man is a legit genius. At an age when most kids are just finishing school, he was making one of the biggest discoveries in all of human history. But he's also a real lunatic. I mean, that's not debatable. He's a nut. In recent years, he's got into a lot of trouble for saying some truly awful, backwards things about women, minorities, pretty much everybody. So he's really not someone you probably want to look up to as a person, despite his scientific accomplishments. And he's also got in much poorer health as years have gone on. His age has advanced a lot. Now, I put this little thing up here just because about a year ago, Jim spent some time in the hospital after flipping his car into the water near his house. This, sadly, was not really a surprise to those of us who have ever spent any time around him, since he always drove about 80 miles an hour minimum down a one-lane winding road in Cold Spring Harbor, and it really was not unusual at any time to see scientists just kind of diving out of the way off the road as his little green classic Jaguar just went roaring past them at, again, way, way faster than he should have been going. And I can guarantee that on the day this was taken, as he was speaking in this lecture hall in the pictures, that car was definitely parked fully up on the sidewalk outside the building. Just right there where it should not definitely be. There are 4 types of bases that give the instructions in DNA. In the same way the English language uses 26 letters to make a huge number of combinations for different words, DNA only has 4 letters in its language, but those letters spell out our recipe book. The 4 bases are adenine, thymine, guanine, and cytosine, abbreviated A, T, G, and C. I probably don't need you to memorize those names, but you definitely need to know the A, T, G, and C. Notice how things are arranged in the bottom picture. The 2 strands, or sides of the chain or ladder in a DNA molecule, are complementary to each other. That means that there's always an A on one side, so there must be a T across from it. And anywhere there's a C, it'll be across from a G. They're only ever found in those pairs, as we see in the top left also. So see how the A and G are a bit bigger than the C and the T? The A and T have 2 rings in their shapes, while the C and T only have 1 ring in their shape. We'll come back to that in a few minutes and see why that's important. So let's match some bases here and try to figure out what's going to go where. We can see now 2 sequences of DNA, one on the left, one on the right. Each one has 2 strands, so we're going to try to match these bases up. On the top left, I see a T. What always has to go across from a T? There's only one thing that can ever go there, and that is going to be an A. Across from G, we will only ever find C. Across from C, in the same way, we're only ever going to find G. And much like the top example, across from an A, we see another T. So notice it was color coded also on the left. The colors are going to be different on the right, so don't get caught up in reds or blues. We see C, A, T, G going down the left side of that strand. So what are our 4 letters on the right going to be? Well, across from a C should always be a G. Across from an A should always be a T. Across from a T is always going to be an A. And across from a G, you always have to have it attached to a C. Now, before we move on, just one more time, take a look at these short DNA chains here. In each pair of complementary bases, we have a bigger 2-ring base with a smaller 1-ring base. So if you notice, the T's and C's only have 1 little ring shape in their structures, whereas the A's and G's have 2 rings. So it doesn't really matter which one's on the left, which one's on the right. As you can see, you can have A on the left or the right of the sequence. You can have G or C or T. It doesn't matter which way they go, but you're always going to have 1 big one with 1 smaller one. This makes sure that you're always going to have the strands be parallel to each other. If you had 2 of the smaller single ring structures, like a C with a T, they would suddenly get very, very close because they only have a small structure each. And if you had two of the bigger ones, like a G and an A, they'd have to push back from each other because they'd be too big to fit next to each other properly. So by having always one small with one big, it makes sure you're always exactly the same distance away from each other, and everything stays nice and parallel, and the sequence and the molecule stays a very nice normal structure. And here's some practice questions from near the end of your notes. Let's take a look at this. So it says a strand of DNA has the sequence AGG-TCA-CAT. And wants to know what is the sequence of its matching strand. So what would be on the other side across it? So I'm just going to draw two little lines here to represent where we've got just the kind of the rest of the sequence goes off in either direction. We don't really care what that is. And on the top left, we have our little matching column here, or matching picture, to show that A is always across from T, C is always across from G, and so on. So the number of A's should always equal the number of T's in a molecule, and the number of C's will always equal the number of percentage of G's in a molecule. So I'm just going to go through and match it up. Right here, I see an A, so I know now that across from that has to be a T. I see a G, and I know that that always has to be across from a C. And the next one's also a G, so it's going to be another C. Across from a T, I have to have an A. C has to be paired with G. A is always paired with T. C again is always paired with G. A is always paired with T. And T across from an A. So it's only going to make sense if it works this way. If anything ever happens, any kind of mutation, where maybe, let's say this A was deleted and some mistake was made, and let's say a C is put here by mistake, your body's really, really good at sending someone to proofread, and very quickly you'd get that little C erased and the proper A base put back in its place where it should be. Question at the bottom. Number 2 says a sample of DNA is measured to contain 30% C's. So what are the percentages of the other bases, the A, T, and G, in this DNA sample? This may seem like a strange question, but all we need to know is one number like this, and we can tell everything else that we want here. So as soon as we know that 30% of the bases in a DNA sample are C's, what does C always have to match up with? What, right here, what does C always have to be across from? Right there, we see that C's and G's always have to match. So if we know that the C's are 30%, that means the G's are also 30% of that molecule. Now we just have to figure out the others by doing a little bit of math. So I see that C is 30% and G is also 30%. So 30 plus 30 is going to give us 60% of our molecule. So the other 40%, 100 minus 60, is going to give us 40% left to work over, play around with. So that means the A and the T together have to add up to 40%. And since they have to be the exact same number, it's just 40 divided by 2. And that's going to give us 20% each for both the A and the T. Simple math. As long as you know one of these letters, how much it is, you can tell everything else really, really quickly. If that doesn't make sense, make sure you email me or come to Extra Help, because that's a very core concept, just being able to do that quick kind of math. So let's work that out if you don't know what you're doing with that. One more like that. Once again, we've got a little table here. We want to fill in these numbers. So I'll just go quick through this chart here. It says that humans have 31% adenine, and if the adenine is 31, we know that thymine also must be 31, because A's and T's have to be the same. I'm going to go down to the cow and do the same thing. If cows have 28% A, they also have to have 28% T. Wheat has 27% A, so it has to have 27% T. And yeast has 31% A, so it has to have 31% T. Going kind of backwards, salmon has 29% T, so it also has to have 29% A. We've now got our outermost columns done. We're just going to do the exact same thing. Humans have 19% C, so they have to have 19% G. Cows have 22% G, so it has to be 22% C. Salmon has 21% C, so it has to be 21% G. I'm going to jump down to the bottom. Yeast has 19% G, so it has to be also 19% C. I didn't bother to do this math here before I started. We see that wheat has 27% each of A and T. If we add up 27 plus 27, that's going to give us 54. So what does that out of 100? We were left with, what, 46% left? That has to be made up of these two numbers right here. So these two have to add up to 46. So we just divide 46 by 2, and each of them is going to be, that was messy, each of them is going to be 23 for G and C. As long as you know one, you can figure out everything else. Let's do a few multiple choice questions together. Number one says, 4 different segments of a DNA molecule are represented below, and just gives you 4 different options here of how DNA would look. It says then there's an error in the DNA molecule in which one? We just have to go through and find something that doesn't make sense. Now remember, you always have to have A going with T and C going with G. You have to have the same amounts of each other, and they have to be across each other. So let's find something that does not look like that. Looking at segment 1, I see T with A with T, G with C, G with C, and C with G. So this one looks pretty good. I don't see any mistakes there. In segment 2, I see a G with a C, a G with a C, a T with an A G with a C, and an A with a T. That also looks fine. That's not going to be our answers there. Looking at my multiple choice answers, segment 1 is not an error, so I'm going to cross that choice off right there. I now know that segment 2 does not have an error, so I can actually get rid of both these choices right here, and it sure looks like segment 3 is going to have to be my answer, but let's double check that. Segment 3, I see a G with a C. That's good. Oh, right there. A with C. That is not how that should go. A should not be across from C, so right there, this is a mistake. I'm just going to double check segment 4. C with G, A with T, T with A, and G with C all look fine. So segment 3 is the only one that's a mistake. That's the one right there that has the error. It's just going through and making sure the letters all match, and segment 3 did not have letters that match and go with each other. Number 2, diagram below represents a section of a molecule that carries genetic information. It's just a picture of DNA. The pattern of numbers represents, so what are these numbers? Right here in the middle, what part of the molecule is here in the middle of the double helix? What do we call those things? If you go back to that structure of the nucleotide, we know that the phosphates and sugars are on the outside, and the bases are the ones that stick into the middle. That's what you got to know here. Order of proteins in a gene. Proteins do not make up genes. Amino acid, that's what's a protein also. That's not going to be an answer here. Sequence of paired bases, right there. Let's just double check choice D. Position of gene mutations, hopefully not, because we don't know about any mutations, but it definitely is bases. That's all we have to know for choice for number 2. And we skipped number 3, went right to number 4. The diagram below represents an incomplete section of a DNA molecule. The boxes represent unidentified bases, so we just have to fill those in first to see what those letters would be before we're going to look at the question. So I see a T across from an empty spot. I know T always goes across from an A. So again, over here, I'm going to put T across from an A. And the last choice, the last spot here, I see an A across from a T. So that's actually 3 of the same kind of pairing right there. And the one that we left blank, C always goes across from a G. So now we filled that in. Let's take a look at the question. It says, when the boxes are filled in, as we already did, what will the total number of bases represented by the letter A, both inside and outside the box be? So how many A's do we have? See, that's all it's asking us. I see one A right there, another A right there, another A right there. So it's 3 A's. That's it. Nice and easy. Make sure your A's match your T's. Make sure your C's match your G's. This is a picture from the original scientific paper published by Watson and Crick to announce to the world their discovery of the double helix. And it's really short. The whole thing is only about a page long, which is kind of insane. There's one paragraph worth looking at, though. This one I'm going to circle right there. It says, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That means the fact that they said A and T and G and C have to go together, and the structure of this makes them think that this whole structure, this helical shape, could give a really easy way for the DNA to make a copy of itself. That's all that's saying. Now, that one sentence right there has been referred to as the greatest understatement in the history of science. That sentence that just so casually says, the shape probably has some effect on how DNA copies itself. That's basically the root of some of the biggest scientific discoveries of the 20th and 21st centuries, continuing to this day. Why and how that is, that's what we're going to do to finish up today. Let's say you have a molecule of DNA in a cell, but that cell wants to divide to make a new cell. So taking one and splitting in half to make 2 new ones. If you don't make a copy of the DNA, then you won't have enough to go around when you split the whole thing in half. So you need to make a new molecule of DNA first. But how are we supposed to do that? Well, the backbone of the DNA is very strong, but the bases aren't held to each other very strongly at all. So it's really easy to kind of unzip them right down the middle of the ladder. And because we know that DNA has to have a and a T together, and the C and G have to go together, the cell is now able to add new nucleotides to these two unzipped strands. And there's only one way that can happen, A with T and C with G. So what's that going to leave us with? It leaves us with two molecules of DNA, each of which is an identical copy of both each other and an identical copy of the original DNA molecule on the left there that we started with. And this is called semi-conservative replication, meaning that each new DNA molecule, you see these daughter strands, we call them, is half original and half new material, as we see in green / this orangey color. So the original molecule just splits right down the middle, and then these new bases just come in and pair up wherever they have to be. The cell has just a supply of A's and T's and C's and G's, and now anywhere there's an A, it'll add a T, anywhere there's a C, it'll add a G, on and on like that, until we get two brand new ones of what looks like the exact same molecule that we started with over there. And they each look identical to each other as well. So that's semi-conservative replication. This is like a real-life cheat code, like a video game. It's so simple of an idea. As long as A's with T's, G's with C, but this and the helical structure of DNA make it possible for everything to happen really easily and quickly in our bodies and everywhere in living things. I want to get you started on this one part of the worksheet just because this is on I think it's the last page of your homework. This is a question that everyone always asks every year when we do this stuff, so I just want to make sure it's clear. On the left side here, we have one original DNA thing, one original DNA molecule. You see it's the two strands, so strand 1 and strand 2. That's just saying when you have your DNA molecule in your ladder, this would be strand 1 over here, this is strand 2 over here, because they have that kind of little break in the middle where the bases are, right? So we have strand 1 and strand 2 right here, and then it shows us with the arrows that this is going to be split apart. We're going to cut it right down the middle here, right in half, so that we can make a brand new copy, or two brand new copies rather, and we're going to see here how this works. So that goes over here now to the things on the right, where it's now opened up, and what was the original DNA strand 1 on the left side here is now right here. And what was the original DNA strand 2 on the right over here is now over here. So just notice these little plus signs right here. These are two separate molecules. So this one original DNA molecule right here was opened up. It split straight down the center here. We unzipped it. And now these right here are the two brand new DNA molecules that we're making from it. So I'm going to just copy what I see on the left and put it onto the stuff on the right so I can go back and fill my molecule. So original DNA strand 1, that's going to be right here. It says right there, copy from left. So I'm going to copy this exact same sequence right here. And I'm just going to write in A and C and T, T, A, C, G, C, C, G, A, T. All I did was copy these letters here from strand 1 and put them right here for strand 1. So let me erase that and go do the other one on the other side now again. This says strand 2, copy from what's on the left here, so I'm going to copy strand 2 on the left too. So I'm going to again write T, G, A, T, G, it looks like a 6, but that's a G, C, G, G, C, T, A. So all I did was take these two complementary base pair strands, and now they are here just across from each other. That being said, that being done, Now I can go in and fill my new letters in. So right here I have an A. I know there would be a T across from it. I know there's a C, so that would be a G. I know there's a T, there should be an A, and so on. On the other side, a T goes with a new A, G goes with a C, and A goes with a T. All you're doing is pairing them up. And really, if you didn't want to do that, if you kind of wanted to shortcut it, you know you're going to get two new molecules of what was over here. So look what we just did. ATCGTA, you just made the brand new one. ATCGTA right here. And same thing right here, ATCGTA. So you made two brand new copies of the original molecule. That's all that's going on here. So now you should hopefully be able to fill in the last sheet of your homework pages. I know this was a much longer video than we've had before, but I hope to not make a habit out of that. I'm figuring 20 minutes each for this chapter probably, though, just because there's a little bit more material here. We do have to kind of take our time to go through it. So go right now and answer the 5 formal questions for today. I'm giving you this video on Tuesday and another much shorter one hopefully on Thursday. The work for both of them will be due next Monday, the 20th at midnight. That's technically 11:59, so it's Monday night, not midnight Monday morning. I was going to do another Teams extra help conference on Wednesday, but I realized I have a full morning of administrative meetings already, so that's just not going to work. I will have one on Friday and another on Monday, though. So you'll have 2 in the meantime before anything is due. Before that, just please email me any questions. As I said before, we're starting a totally different kind of material here, so it's important to be sure that everything makes sense at the beginning. Otherwise, things can really easily get away from you pretty quickly. So otherwise, stay safe. I'll talk to you all soon.